Okay.
All right. Good morning, everyone. Hope you all had a great weekend. We just finished up with Unit 1, finishing our conversation there on soils. And we'll pick up a little bit later on talking about soils a little bit more and the things that we do to soil. So we're looking mostly at the natural properties. I've got part 1 of the lab up, which is basically just doing the soil texture test. That's with the mason jar, and there's a pretty good video that guides you through that process on Canvas. So be sure to get on and conduct that lab, looking at soil texture. And there will be a part 2 of it that deals with surveying your soil and examining the texture or the structure a little more. But we're going to go ahead and move on to the beginning of Unit 2. I set all of the quizzes as due tomorrow at 11:59 p.m. That's Tuesday, October 1st. So if you can get on there and finish up those quizzes, then on Wednesday we'll have a review. And actually we're pretty close to midterm time. We might actually do our midterm a little bit earlier than the actual midterm itself. So just be getting ready for that. Make sure all the quizzes are in, labs are completed up to this point, so we can start really getting into Unit 2. So the first part of Unit 2 here is starts with looking at plant parts and kind of the structure of higher plants. And this beginning of Unit 2, I will say, is does get little into the botanical parts. And I've kind of weeded it down a little bit to where I think we're just hitting up the most crucial plant parts and their functions from a practical standpoint. But there is some science, of course, that we should understand. So just bear with me here, and we'll kind of take our time on the next couple of chapters instead of trying to just rush through them. So let's go ahead and get into Chapter 6, the structure of higher plants. All right. So going back to kind of the beginning, the nitty-gritty of it, we do need to study plants from a structural standpoint to understand how we can really optimally produce them. And it's important to think about how they're classified, the taxonomy, the botany, all of that kind of goes together again from this kind of, I should say, practical standpoint. Starting off, just thinking about the taxonomy. Almost all of our major food, fiber, wood, and even the ornamental plants belong to 2 main classes of plants. Gymnosperms, which we look at the narrow-leaved, evergreen conifer trees, and angiosperms, which are usually broad-leaved and flowering plants. And these 2 classes, if you will, are actually referring more to the phylums of the plant. So if we look at just the way that living organisms are classified, we start off at the kingdom level, of course, the most general. And of course, they are in the plant kingdom. All plants are in the plant kingdom. The next step down is the phylum. And we look at the gymnosperms as the coniferophyta, the conifers, and the angiosperms as the magnoliophyta. So they say 2 classes, really 2 different phyla of plants, the most broad general sense. The angiosperms are the most successful plants and have colonized, basically every continent. They are found all over the world. They're the most successful group of plants because of their coevolution with insects mostly. And we further, break down the angiosperms into the actual class level, looking at the monocots and the dicots. And we'll look a little bit about how to differentiate those. But the main thing is that the monocots, when the seed germinates, they have one cotyledon, which is the seed leaf. And inside of the seed, we actually have an embryo. And what that seed does is it basically sends out the radical, this little baby root, pretty much, taproot, and it sends out the cotyledon. And that cotyledon has all this sugar kind of fat reserves stored there, and is actually the bulk of a lot of different types of seeds. So when it comes out, monocots have one blade, one leaf coming out of the ground. Dicots have two cotyledons, two seed leaves. So let's just look at an example of a monocot. Probably our most common monocot crop, at least in the ag world, looking at corn, Zea mays, a monocot. So as it pops out, you can kind of see here in the seed here, the corn actual kernel itself. In there, we've got this portion that is the radical, and then this portion that is that seed leaf itself. And together, those make up, a bulk of the seed itself. And the endosperm being one of the places where there's also food reserves stored other than just the cotyledon. So when it's planted in moist soil, the seed begins to imbibe or absorb water from the soil solution, and that signals it to break dormancy, sends out that radical, that primary root, and that one cotyledon. So the radical is the taproot pretty much. And the plumule here, kind of looked at as the top of the radical, is the primary shoot. And as these two grow in opposite directions, in the beginning, they kind of are fairly even in growth. Roots are rapidly expanding to be able to absorb nutrients. Leaves are rapidly developing so they can start to photosynthesize. And in this period of rapid growth, it's fairly even above ground and below ground. The radical will continue to grow down through what is known as the coleorhiza, and then from that, the primary roots develop, and the secondary roots branch off, getting more of these root hairs, which is actually where the plants are getting that nutrition from. A mature corn plant can develop roots up to 6 feet long. And of course, the above ground part of the plant can be anywhere from, seen some short corn varieties, 3 feet or so, all the way up to some corn varieties getting up to about 12 feet. So quite a substantial root system, as well as a large plant above ground. In corn and also many other monocots, we also have this whole concept of adventitious roots, which are basically just roots that come out from any point other than the main root. So you can kind of see, and I'm sure you all have seen this in corn, that these, first they kind of make these little nodules near the node that pop out of the stem. So it's rather than coming from the primary root, the coleorhiza and the radical, they're coming out from the stem itself. And these are called anchor or brace roots in corn because they're just giving added support to support that tall plant so it doesn't fall over. We have adventitious roots in a lot of other plants too. They don't just occur in monocots, but it's usually as a response to high moisture levels. So you can imagine that near the soil surface, they would be developing. Whenever the corn begins to grow, the corn plant or other monocots, initially the plumule, that shoot, is protected by this coleoptile, which basically just envelops that main stem and kind of has this this sheath around it to protect these young developing leaves until they're able to grow out and start to photosynthesize themselves. When the plant reaches a given size, when it gets to sexual maturity, and that's depending on the plant, it's measured by different factors. In corn, it's usually a set number of leaves. The female flowers, or the pistillate flowers, the female part is the pistil, begin to emerge. And in corn, we look at this as the ears of corn, and the pistils themselves are actually those silks at the tip coming out of that ear of corn. And then a little later on, we'll have the male flowers, the staminate flowers, the stamen being the male part that produces the pollen, emerging at the very top of the plant, and they call that the tassel in corn. So here's just an image on A there showing the top, the tassel, and B, the silks, the pistils, and then C, what happens when those silks, each of those pistils intercept a grain of pollen. Each of these pistils is connected to each kernel, which at that stage is just an ovule, like a little baby ovary, and each one of those has to eventually meet a grain of pollen, to be fertilized and to develop into an actual corn kernel that we eat. So then what happens, often for corn, we might eat them fresh as sweet corn, or we might leave it on the plant to dry. Either way, the ovary develops into a fruit. We call it a kernel, but in the botanical world, it is a caryopsis, which encloses the true seed, what will become dormant and eventually become a new plant. After these kernels mature and dry, we harvest those seeds, store them over the winter to grow them again, or we feed them to animals in many cases. Maybe we process the plants. Let's take a look at the dicot. It's a slightly different process. So we'll look at the, we're looking at a grain here, corn, and then a legume in the dicot world, the bean. So again, after that bean seed, which is dormant at that point, it's still alive, but it's just in stasis, right? After it is put into moist soil, it begins to suck up water and starts to swell, and then the seed coat is going to be broken by the emerging radicle on one end and the plumule on the other end. And in the bean, it's called a hypocotyl, and this is what we look at as the hook. Basically, it's part of the plumule, but it's not the top part of it. So it emerges first above the soil, and then it's able to kind of spring up and push those cotyledons out. And of course, in a bean seed, we have the two cotyledons because it's a dicot. If you look at this image here on B, you can see that actually those cotyledons are, the majority of the seed there. So that hypocotyl is the area below the cotyledons and above the radicle. And then it starts to emerge leaves. And often in dicots, monocots as well, but much more in the dicots. As the first sets of leaves emerge, they look totally different. The cotyledons don't really resemble the leaves as much. The primary leaves don't look like the mature leaves that later on develop. And so notice here, the primary foliage is a single leaf, a simple leaf, whereas the next leaves to emerge, the trifoliate, they are compound leaves. So it changes in morphology as it grows. Between cotyledons and really at every node, we have a growing point, and that is called the apical or shoot meristem, depending on where it's at. The apical meristem is at the top. The shoot meristem will allow it to branch out in either direction. So the important thing about the meristem is this is where growth is basically occurring, where it's initiated. So of course, as they grow, that meristem is actually kind of leading that growth in both directions, going up as well as out. The cotyledons themselves, once that energy is absorbed by the plant and the plant is able to photosynthesize on its own, they're just going to shrivel up and drop off. So as the bean plants are growing, that meristem is where these cells, the stem cells, right, are basically becoming differentiated into different plant parts. So whether they're making new leaves, or if they're making stem cells, literally, or if they're making flower buds and flowers. In beans, and this is not all dicots, in fact, not a good portion at all anyway, but in beans, the flowers are actually self-pollinated. So before the flower even opens, it's fertilized itself, which makes it really easy to save seed from. The fruits are going to develop into a pod, which contains the maturing ovules that turn into new beans. And as the plant grows, we can, harvest that pod early as a green bean before the seeds have developed that much and before the pod gets really fibrous, or we can allow them to mature and dry within the pod for seed. Let's take a look at the cell. So the cell, of course, is the basic primary structure of all living things, and in plants as well. And the cell is where, this primary unit, where it's going to develop multiple cells into plant tissues, which then develop into organs to make the full growth of the plant. We look at cytology as just this kind of subfield of biology where we're actually studying cells and their functions. Cells can actually vary greatly in size. The smallest cells being micrometers that you can never see by microscope. Even a regular microscope, we have to look at an electron microscope to see the smallest cells. Largest cells, though, can be up to several centimeters long. We could see them with the naked eye. Kind of crazy. So cytology comes from the term cytoplasm. And the cytoplasm is just basically this kind of juice in the cell. It's this, plasma, technically, where we have all of this cellular material kind of being distributed and moving within the cell. So just talking about kind of general cellular structure, we look at basically two different types. We have prokaryotic cells, which are kind of the primitive cells, and they don't have any separate subcellular units. Everything's just kind of flowing in there. So prokaryotic cells are often found in really simple organisms like bacteria, and some maybe that we think of as a little more advanced, like blue-green algae that can even photosynthesize a little bit. Eukaryotic cells are those that have compartments and are bounded by some sort of membrane, that each organelle within the cell has specialized structures and functions. So we can look at the organelles here, the most important being the nucleus, the brain of the cell, right, where most of the DNA is stored, not all of it, but most of it. The mitochondria, which we refer to as the powerhouse of the cells, the plastids, the microbodies, the vacuoles, the endoplasmic reticulum. These are all important to know about, but I'm going to kind of gloss over them here. Just know that these are the components of the cell in a eukaryotic cell. Eukaryotic organisms are those that are made up of multiple cells, whereas the prokaryotic organisms are single-celled organisms. All plant cells are eukaryotic cells. And here's just a close-up looking at cellular structure. And one of the things that is unique about plants is they have this thick, kind of rectangular shaped cell wall, which basically holds that cytoplasm in. And speaking of cytoplasm, you know, here it is. Basically, you can kind of see it throughout the cell. It's this juice that everything's kind of floating around in. Point out the nucleus here being this large structure, the center of the cell where most of the DNA is stored. And that's kind of, I'm not going to dwell on that too much. So again, on the cytoplasm, it is this kind of viscous fluid, and within it are matrix proteins that are bounded by this plasma membrane, which is semi-permeable, which allows nutrients, molecules to come in and move throughout the cell. The flow of the organelles and basically the cytoplasm itself is called cytoplasmic streaming. And it is clearly visible in active leaf cells under just a regular light microscope. And it's part of what we see when the cells start to reproduce and divide too. It is important to note that in the cytoplasm, there is a little bit of DNA as well. The endoplasmic reticulum is important because that's where proteins are synthesized, and where we actually have the creation of chlorophyll. And so chlorophyll is a type of chromoplast, which contains the plant pigments. And within the chlorophyll are chloroplasts, which are responsible for photosynthesis. And as we start to see, now and in the coming weeks, as the nighttime temperatures lower, we think of light, right, as this very important thing for plants. An electromagnetic radiation coming from the sun, which is broken down into these different wavelengths that correspond to different colors, right? Red, orange, yellow, green, blue, indigo, and violet. The chlorophyll actually reflects green, but in the chloroplasts, the other colors are stored. So when we get those low nighttime temperatures, it signals the leaves to begin to senesce. Chlorophyll dies, so it's no longer reflecting green, and it reveals the true pigments of the chloroplasts, the yellows, reds, scarlets, purples that we see in autumn. It's kind of neat. But basically, this photosynthesis process is occurring there in the chloroplasts and creating chlorophyll. The mitochondria, what we say in biology is the powerhouses of the cell. They're the sites of respiration. And they're also involved in protein synthesis. So proteins are made in the endoplasmic reticulum, but then they're further synthesized in the mitochondria to help to produce energy, basically. And one of these energy-rich compounds we look at is adenosine triphosphate, ATP. It's basically just an energy carrier, how the plants move energy throughout the cell and throughout the plant itself. So I mentioned that the nucleus is home to most of the DNA, and that is stored on chromosomes within the nucleus. So we look at the deoxyribonucleic acid, the DNA, which is just these long strands that are basically more advanced forms of proteins that carry our genetic information that code for all of the cellular functions and the differentiation of those cells to become the different parts, as well as for reproduction. So we have genes being the subunit on the chromosome. And the genes are transcribed from the DNA to say this is what that plant is and what it's going to look like. And as I mentioned, we also have some DNA in the mitochondria, the cytoplasm, and in the chloroplasts. So these also have some role in the heredity process of moving DNA, but not nearly as much as just the chromosomes in the nucleus. Vacuoles are actually a large portion of cells, and in a mature cell could be up to 90% of the volume. And the basic function of the vacuole is to serve as a storage reserve for water and salts. And it's kind of like the liver of the plants. It's processing toxic products as well. So we also see such things as pigmentation in vacuoles. And there's a membrane around the vacuole known as the tonoplast that controls the flow of water and the maintaining of the turgidity of the cell. It gives it that rigid cell structure. In the cell wall. The cell wall itself is not alive. It's just kind of, in plant cells, this rectangular structure that is there just to protect everything and give it some rigidity. And it is made of cellulose as well as some pectic substances and lignins, which we'll talk about later on. There's also the potential for suberins and cutins depending on the type of plant. And these things are basically just compounds that the plant uses to create the rigidity of the stems. The suberins, especially for wounding of plants, these chemicals are utilized to heal wounds in plants. Cutins are in the waxes of the leaves, the cuticle, right? Cutins, which basically are there to protect the plant and the cells inside of the plant. So as I mentioned earlier, the cell is the basic unit, but when we have all kinds of cells being organized, form a similar structure that has a collective function, we call those tissues. So think of tissues as kind of the basic structural component of the organs. So cells make up tissues, tissues make up organs. It's the same as it is in humans, but the organs are different, right? We have leaves, we have flowers, fruits, stems, and roots. What we're thinking of as kind of the basic divisions of tissues in the plants. And we have those that are vegetative, we look at, that are kind of undergoing this regular cell division, are roots, stems, and leaves, the vegetative parts of the plant. The flowers, fruits, and seeds being the reproductive parts of the plants that are undergoing meiosis, the sexual reproduction and cell division. We have two basic kinds of tissues in plants, and we mentioned the meristem earlier. The meristematic tissue is where cells are actively dividing. And within them, there was this controversy some years back talking about stem cells, right? So this is kind of what these meristematic cells are. They're undifferentiated cells. So they're a basic cell that is then given a chemical or a hormone or a different set of proteins that turns it into a particular type of cell to develop into a tissue or an organ. So the meristem is where that takes place. And actually in plants, we have the apical meristem, which occurs at the top of the plant as well as the bottom, where the cells are actively dividing to grow upward, downward. And there's also a meristematic area that allows for lateral growth as well. So here we have the shoot apical meristem, the top of the plant, the terminus of the above ground portion of the plant. So this is responsible for producing new buds, new leaves along the stem. And then we have these axillary buds that have these secondary meristems that are producing branches. The apical meristem, though, is the top of the plant, what we consider in many plants to be the leader. That's also where a lot of these chemicals, especially the hormones, are kind of concentrated and move throughout the plant to assign different tasks to different cells, dividing in the other meristems. So we also have the root meristems at the ending of the roots, of course. So the growing point for the root system. And, that begins as the radicle, where in all plants it kind of actually starts as a taproot with the radicle, but in some plants they quickly switch to more fibrous root systems, like in this grass picture here, versus other plants that have a very well-defined taproot, like a carrot, for instance. A lot of biennial plants have these root crops, right? So those that don't have the dominant taproot, at least later on, are referred to as fibrous root systems. And we have that meristematic zone and the roots that, is basically dividing to say you're going to be a root hair, you're going to be a root cap, etc. We also have the lateral meristems that are producing the secondary growth, kind of the thickening of the plant stem and the branching off of it as well. So what we're looking at here is kind of more like in a woody plant here on the right, where we have cork and cork cambium, which becomes the bark of a plant, which is a little bit different than an herbaceous plant. So this is another way to kind of divide higher plants, is whether they are herbaceous and woody. The difference being that the woody plants have this kind of secondary xylem, which we'll talk about here in just a moment. The vascular cambium is this section of blue in the image here. And in the woody plants, actually really the main part that is alive. We get a little bit of life kind of going in both directions. But the xylem is actually mostly dead cells. So here it's a meristematic region where it's producing on one side the phloem, the cortex, the cambium, the epidermis, and on the other side it's producing new xylem. So it's making the outer part of the plant, it's thickening the plant kind of in both directions. So here's looking at the stem, a cross-section of the stem of a woody plant. And we see this in some herbaceous plants as well, the woodier perennials. So the stem, the girth is actually increasing, and it's by these lateral meristems. And in this case, we can use these alternating rings of growth as a way to age the plant in woody plants. So this is where we get coring to figure out what the age of plants are actually. We'll take a horizontally extracted core of a woody plant and count these rings, because each ring is basically where that cambium quit making that year's xylem and phloem on the other end. Epidermis, just like, is a simple tissue, just like in humans. We think of it as the outside, the skin, basically. The difference in plants is that it's covered with this waxy substance known as cutin, which basically is just reducing water loss. And we can see in this microscope image the stomata. So these are very important for that process of transpiration, the exchange of gases from the leaves. So these are on the underside of the leaves, and around these stomata are these guard cells that are triggered by, certain chemicals to open and close throughout the day, and in the evening to allow for the exchange of gases, basically to release oxygen and respire, and to take in carbon dioxide so they can continue to photosynthesize. So they're like the pores in our skin in a way, except they're just on the underside of mostly of leaves, and some plants they're in the stem as well. We also have in some leaf epidermal cells hairs or trichomes. And these trichomes are basically just areas where, again, it's kind of for protection of the plant, but they can also store some chemicals as well. And that's often for protection of the plant, but it could also be for to attract a pollinator, for instance. So here are just some of these structures magnified. You can kind of see the hairs themselves, and the trichomes, these kind of bulbous structures. And then we have the permanent tissues of the plants that are a little bit more complex. So first we have xylem, which is structurally very complex tissue. And the xylem is basically just used to conduct water and the dissolved minerals that are sucked up by the roots to all the different parts of the plant. So it's mostly moving water and nutrients up the stem of the plant. And so we see cells that are specialized within the xylem to act kind of like the circulatory system in humans, right? So we have vessels, tracheids, and fibers, and parenchyma that are involved in the movement of water and nutrients. The phloem is basically the tubules that are comprised of these sieve tubes that conduct food and the metabolites, and these can also move up and down the stem. So we generally think of phloem as moving downward because it is where we're moving the metabolites that are produced in the leaves and the chlorophyll and moving them to the other parts of the plant. And for instance, we need nutrients that are molecules that are produced in photosynthesis to create more roots. So they're moving that energy down. But the phloem can also move energy from old leaf cells to new leaf cells. So it moves a little bit in both ways. For simplicity's sake, we'll think that phloem goes up, xylem goes up and phloem goes down, but really they kind of operate in both directions. So phloem food, xylem water, and here's just a cross-section of a root. And you can actually see from the root, we have the root hairs. And the root hairs are what is actually absorbing water as well as the dissolved nutrients in the soil solution. And they take that actually in through these kind of guard cells and different layers into the xylem and into the phloem to basically move these nutrients and energy upwards and downwards throughout the plant. You can also see here that in the roots we have what they label here as the quiescent zone, and behind that quiescent zone is the actual meristematic region of the roots, where the cells are actively dividing, some of those being told to become the root cap. The root cap is what is actually pushing that primary root through the soil. It can actually have these lubricants, as we mentioned, that it leaches out through the root cap, allowing it to press through these pores in the soil to get to areas where the root hairs can absorb water and air. So for that soil solution, that which is just water flowing through the soil, to actually enter into the inner tissue of the root, it has to pass through these semi-permeable cell walls. So there's this kind of pathway that allows the root hair to move this water and the nutrients dissolved in the water through cell walls, and finally, eventually, into the xylem and phloem. It's a complex process. We just need to know that it happens, and why it's important to have particular conditions that allow it to do that. We mentioned the adventitious roots earlier, those that form from any place other than, the radical and the regular roots themselves. So the big thing for adventitious roots is that we're actually using them, and they're kind of initiating from this cambium layer, kind of in between these vascular bundles that are, again, kind of like the veins of the plant. They're, moving these substances throughout the plant, circulating these substances. The adventitious roots come kind of from between these bundles. And we use that adventitious root production as a way to propagate plants asexually, to clone them and make them into cuttings. They don't have to just come from stems. They can also come from leaves in some cases, and also from old root pieces. So just looking at the cross section of a stem here, and this is of a woody plant because we can see we got that cortex. We have the primary xylem. We also have secondary xylem here as well, which is not labeled, but that secondary xylem is what is basically the wood of the plant. And again, that's all basically, dead cells. So this would kind of be our vascular cambium here, and where it's being differentiated in that meristematic region into xylem and into phloem. Let's look at a few different types of stems here. First, we got a rhizome, which many people think of rhizomes as roots, but it's actually just an underground horizontally growing stem. So we use these rhizomes as a way to propagate plants, because you separate a rhizome, and it's, as long as it has a budding section on it, it will develop into a new plant. So bananas, canna lilies, irises, and bamboos, these are examples of plants that reproduce through these rhizomes. We also have stolons that are stems that are kind of growing horizontally, except they at least begin above ground. Some of them will kind of start to go underground as they develop what some people call runners. So the classic example of those with stolons are strawberries. Some ferns do it. Bermuda grass does it. Horizontally growing above ground. We also have the bulbous plants that have these specialized structures. Again, we think of these as being roots, but the roots are always, below this particular point here, this meristematic region. So this is an example of a corm, a thickened compressed stem growing underground. And we have these bulbing plants such as gladiolus, crocus, that produce this type of stem, the corm. Bulbs are, kind of, I think of a corm as a type of bulb, but a bulb is actually a specific type of compressed underground stem. Again, these are storing energy. They're not really roots though, right? Their roots come out from the bottom of them. So most bulbs have these scales on them that are basically just the storage leaves. And then later on, they become the leaves of the plant. So we have onions, tulips, examples of these bulbous plants. We also have tubers, which are generally growing underground. They're in large, fleshy terminal portions of underground stems, such as in the potato, right? So it's a tuber, again, a storage reserve for energy of the plant. Sometimes in plants, I mean, the stem gives rise to buds and leaves, but in some cases, like in cacti specifically, the stem and the leaves are actually kind of merged. I mean, they're pretty much the same organ. And then they have the spines that are modified forms of a leaf. But the leaves and the stems, you know, where the leaf meets the stem, we have the petiole, which is like the leaf stem itself. But that is what we refer to as a node. And the pattern, position, and shape of the leaves are going to be influenced primarily by the genotype of the plant, the genetics of it, but then also by the phenotype or the environment that causes them to be morphologically different based on the environment. So going back to our monocots and dicots, on leaves, monocots have kind of a long, narrow, usually strap-shaped leaves that have a parallel veination. So the veins are going up and down the leaf. The leaves of the dicotyledonous plants, the dicots, vary a lot in size and shape. So we can have some that are almost strap-like in some ways, but the big difference is that their veination occurs in nets. So it starts from a single point and radiates outwards. And pretty much all the leaves on here, except for this linear leaf, are dicot leaves, because you can see they have that net veination instead of that parallel veination. They also have different arrangements of the shape of the base of the leaf, the margin of the leaf, and these are ways that we use to identify plants and group them together. How these different shapes, nets, the margin of the leaf are developed. All right, so we mentioned transpiration again, talking about the water cycle, but also it's a very crucial part for the growth of plants themselves. So we have that as moving water throughout the leaf. So it helps to regulate the leaf's temperature, and it's also allowing the straw effect basically to conduct water throughout the stem. If we didn't have water vapor actually leaving the leaf through the stomata and transpiring out, then we couldn't continue to pull water up from the roots. So some plants, of course, have a very thickened cuticle around them to help prevent water loss. Right now, if we got a little bit of rain the other day, but we're still pretty dry here. We're starting to see plants beginning to wilt. If you notice, though, those that have this thickened cuticle, they're not wilting, right? Because it's holding that water in there. All right. When we're looking at the leaf, we look at the blade, which is the flat, thin part of the leaf. The petiole, which I mentioned, is attaching the blade of the leaf to the stem of the plant. And in most plants, there's also this stipule at the base of the petiole. It's kind of a protective organ. The way that the leaves are attached are also important for identification. So if they're attached directly and we don't have a petiole, that's called a sessile leaf. In terms of the identification of plants, and I think we'll kind of cut our conversation here, we look at the leaves as either being classified as simple or as compound. So referring back to the bean, the simple leaf is like the first primary leaf. It's just got one leaf blade, that flat, thin part. A compound leaf has multiple leaflets. So if you follow that one single petiole coming from the stem, it's actually got multiple leaf blades. So in that bean, we look at it as the first leaves as primary leaves being simple, and then the more mature leaves being compound. So we have to recognize that that changes in the development of the plant, as well as it just changes with the environment too. All right, we'll get into this, the other parts of the leaf later on Wednesday. Anybody have any questions about just those basic plant parts and cellular structure? All righty, well, if you all could please go on there and finish up the quizzes up until now by tomorrow night, 11:59 p.m. So we can go over them on Wednesday and get a little bit closer at least to having our midterm exam. All right, we all have a great rest of the day and beginning of the week.
Okay. Bye, everybody. See some of y'all in a minute.